transport, how the transportation takes place. Uh, so let's read the understandings. Particles move across membranes by simple diffusion, facilitated diffusion, osmosis, and by active transport. The fluidity of the membrane allows the material to be taken into cells by endocytosis or released by exocytosis. Vesicles move materials within cells. The application and skills involved here: the structure and function of the sodium potassium pumps for the active transport and the potassium channels for the facilitated diffusion in axons. Tissue or organs to be used in medical procedure must be bathed in the solution with the same osmolarity as the cytoplasm to prevent osmosis. The skill here is estimation of the osmolarity in the tissues by bathing sample in the hypotonic and hypertonic solutions. And guidance is osmosis experiment are useful opportunity to stress the need of accurate mass and volume measurement in the scientific experiment. So here comes passive and active transport. There are two general type of cellular transport: the passive transport and active transport. As mentioned previously, the passive transport does not require energy in the form of ATP, but the active transport does. The so Passive transport does not require energy in the form of ATP, but active transport does. Okay, remember this. Passive transport occurs in the situations where there are areas of different concentration of the particular substance. Movement of the substances occur from an area of higher concentration to an area of lower concentration. Movement is the said to occur along concentration gradient. When active transport occurs, the substance is moved against a concentration gradient, so energy expenditure must occur. Passive transport, diffusion, and osmosis. And the figure here shows chemical diffusion. Chemical diffusion, like note how sugar molecule disperse from the area of higher concentration to the area of lower concentration. This is a lump of sugar. When we drop it in this beaker filled with water, and these sugar molecules expand away, they are like separated and they mix up here. So this is how this diffusion takes place. So now diffusion is only one type of passive transport. Particles of certain type move from a region of higher concentration to a region of lower concentration. However, a living system diffusion often involves a membrane. For example, oxygen gas moves from the outside a cell to inside the cell. Oxygen is used by the cells when its mitochondria carry out respiration, thus creating a relatively lower oxygen concentration inside the cell compared with the outside cell. Oxygen diffuses into the cell. As the result, the carbon dioxide diffuses the opposite direction to the oxygen because carbon dioxide is produced as a result of mitochondrial respiration. And here it's another type of diffusion, which is facilitated diffusion. Facilitated diffusion is particular type of diffusion involving a membrane with the specific carrier proteins that are capable of the combining with the substance to aid its movement. The carrier protein such changes shape to accomplish this task, but does not require energy. It should be evident from the explanation that facilitated diffusion is very specific depending on the carrier proteins. The rate of facilitated diffusion will level off when total saturation of the available carriers occur. Okay, so an example of a disease involving facilitated diffusion is cystinuria. This occurs when the protein that carries the amino acid cysteine is absent from the kidney cells. The result is a build up of the amino acids in the kidney, resulting in very painful kidney stones. Okay, osmosis. Osmosis is another type of passive transport. Movement occurs along a concentration gradient. However, osmosis involves only the passive movement of the water across a partially permeable membrane. The partially permeable membrane is one that only allows certain substances to pass through. A permeable membrane would allow everything through. So a concentration gradient of the water that allows the movement to occur as the result of the difference between the solute concentration on the either side of the partially permeable membrane. A hypertonic, which is hyperosmotic solution, has a higher concentration of total solutes than a hypotonic, which is hypoosmotic solution. Water therefore moves from a hypotonic solution to a hypertonic solution occurs as the partially permeable membrane. which we can see in figure 1.18. If isotonic solution occur on the either side of the partially permeable membrane, no net movement of water is evident. The table 1.7 summarizes diffusion and osmosis passive transport occur across cellular membrane. So the diffusion and osmosis. The type of passive transport are simple diffusion, facilitated diffusion, and osmosis. And description of membrane is substances other than water move between the phospholipid molecules as through proteins that possess channels. And in facilitated diffusion, non-channel protein carrier change shape to allow movement of the substances other than water. Osmosis: only water moves through the membrane using aquaporins, which are proteins with specialized channels for water movement. Here we can see in the diagram the partially permeable membrane between the two solutions of the different osmotic concentration allows water molecules to pass from the hypoosmotic solution to the hyperosmotic solution. So this is the urea molecule in the square shape, and these circles are the water molecules, and this is hypoosmotic solution which they are floating. And so we can see the direction of movement and hyperosmotic solution. This is a partially permeable membrane. So size and change. The size of the and polarity of the molecules determine the ease with which the various substances can cross membranes. These characteristics and the ability of the molecules to cross membranes are arranged along a continuum like this. Small non-polar molecules cross membrane easily, but the large and polar molecules cross membrane with difficulty. How easily a substance can move across a membrane passively depends on the two major factors, the size and the charge, how much energy they have. So substances that are small in size and non-polar will move across a membrane with substances that are polar, large in size, both do not cross membrane easily. For example, small non-polar substances that are gases such as the oxygen, carbon dioxide, nitrogen, ions such as chloride ions, potassium ion, and sodium ions have the great deal of difficulty crossing membrane passively, as do large molecules such as glucose and sucrose. Molecules such as water and glycerol are small, uncharged polar molecules that can cross membranes fairly easily. So here is some practical example that you can read about the kidney dialysis. When person suppose a person's kidney, both the kidneys are damaged, and so he is put on dialysis. He is not able to move properly, and he can't do anything. So this is very interesting. You can read it and challenge yourself with these. Here is an experiment. So active transport in the cell. As you will remember, active transport requires work to be performed. This means energy must be used, so ATP is required. So ATP, the active transport involves the movement of substance such as the concentration gradient. These process allows a cell to maintain interior concentrations of the molecules that are different from the exterior concentrations. Animal cells have a much higher concentration of the potassium ions than their exterior environment, whereas sodium ions are more concentrated in the extracellular environment than in the cells. The cell maintains this condition by pumping potassium ions into the cell and pumping sodium ions out of it. So, along with the energy, a membrane protein must be involved for the process to occur. So, sodium potassium pump. Let's see mechanism of actively moving sodium potassium ions. The sodium potassium pump has 5 stages, and this is the figure showing the pump. A protein in the possible bilayer opens to the intracellular side from this side and attaches 3 sodium ions into it. So a specific protein binds to the 3 intracellular sodium ions, and a binding of sodium ions causes phosphorylation by ATP. ATP has 3 attached phosphates. When it carries out phosphorylation, uh, one phosphate is lost, resulting in a 2-phosphate compound called the ADP. 'ATP'. ATP and ADP are discussed in more detail in chapter 2. So these are the of the figures that you can look at. And the phosphorylation causes the protein to change its shape, thus expelling sodium ions to the exterior. Two extracellular potassium ions bind to different regions of the proteins. This causes the release of phosphate group in this figure as they are showing. And the loss of phosphate group restores the protein's original shape, thus causing the release of the potassium ions into the intracellular space. The sodium potassium pump shows how important inactive specific proteins are in the active transport of the particular substance. It is also clear how ATP plays a crucial role in the active transport. Endocytosis and exocytosis. Endocytosis and exocytosis are processes that allow larger molecules to move across the plasma membrane. Endocytosis allow macromolecules to enter the cell, while exocytosis allows the molecules to leave. Both the process depend on the fluidity of the plasma membrane. It is important to recall why cell membranes are fluid in consistency. The phospholipid molecules are not closely packed through together, largely because of the rather loose connections between the fatty acid tails. It is also important to remember why the membrane is quite stable. The hydrophilic and the hydrophobic properties of the different regions of the phospholipid molecules cause them to form stable bilayer in the aqueous environment. Endocytosis occurs when a portion of the plasma membrane is pinched off to enclose macromolecules and particulates. This pinching off involves a change in the shape of the membrane. The result is the formation of vesicle that it results in the formation of vesicles that then enters the cytoplasm of the cell, and the cell of the membranes reattach because of the hydrophobic and the hydrophilic properties of the phospholipids and the presence of water. This could not occur if the plasma membrane did not have a fluid nature. Example of endocytosis include phagocytosis and pinocytosis, the intake of larger particulate matter, and it's the intake of extracellular fluids. So exocytosis is essentially the reverse of endocytosis. So the fluidity of the plasma membrane and the hydrophobic and hydrophilic properties of its molecule are just as important as the endocytosis. One example of the cell exocytosis involves a protein produced in the cytoplasm of the cell. Protein exocytosis usually begins in the ribosomes of the rough endoplasmic reticulum and progresses through a series of the four steps which are outlined below until the substance produced is secreted to the environment outside the cell. Protein produced by the ribosome of the rough endoplasmic reticulum enters the lumen and in the inner space of the endoplasmic reticulum. Protein exits the endoplasmic reticulum and enters the cyst side and faces the Golgi apparatus. A vesicle is involved. As the protein moves through the Golgi apparatus, it is modified and exits the transphase inside a vesicle. The vesicle with the modified protein side moves to and fuses the plasma membrane. This results in secretion of the content from the cell. So this figure shows how the Golgi apparatus does its function. Please have a look. We have example of phagocytosis when pancreas cells produce insulin and secrete it into the bloodstream to help regulate blood glucose level. Neurotransmitters are released at synapses in the nervous system. So example of exocytosis. The fluidity of the plasma membrane is essential to allow fusion and subsequent secretion of the vesicle content. At this point, the vesicle membrane is actually a part of the plasma membrane. So here we have some beautiful, nice questions that we can practice. Okay. So bye bye. Have a nice day.